There are a number of very promising new agents being developed for HER2-positive metastatic breast cancer, several of which we have recent data with strong efficacy profiles. Trastuzumab deruxtecan is one of those, but at San Antonio this year we also heard about two additional agents. One is tucatinib. This is a tyrosine kinase inhibitor that inhibits HER2. It's different than other kinase inhibitors that we've been using, including lapatinib and neratinib, both of which not only block HER2, but also block other HER family members, such as EGFR. By doing that, they cause toxicities related to EGFR inhibition, mainly diarrhea and rash. Tucatinib is different because it only blocks HER2. It has substantially less diarrhea and rash compared to these other drugs. It was recently tested in a study called HER2CLIMB, which enrolled patients with HER2-positive advanced breast cancer that had previously progressed on not only trastuzumab and pertuzumab, but also T-DM1. In this study, patients were randomized to capecitabine and trastuzumab with either tucatinib or placebo. And what was interesting about this trial was that it also allowed patients who not only had treated brain metastases, but also progressive brain metastases. And this is probably the first large phase 3 trial that allowed patients with treated brain metastases. And the reason it did that, and I'm sorry, not just treated brain metastases, but progressive brain metastases, the reason it did that is because tucatinib crosses the blood-brain barrier quite well. So it was hoped that tucatinib would be able to prevent the progression of brain metastases. And that was seen very well in the data that were presented at the 2019 San Antonio meeting, that the addition of tucatinib to trastuzumab and capecitabine led to a very impressive improvement in progression-free survival with a hazard ratio of 0.54, and interestingly, a substantial improvement in overall survival as well. They also looked at the patients who had brain metastases at baseline, and the benefit in terms of progression-free survival was very impressive with a hazard ratio of 0.48. So this is a drug that clearly has substantial benefit, not only in the overall population of HER2-positive pretreated metastatic disease, but also in patients who have brain metastases, which is a common problem in HER2-positive disease. So I think this is a drug that looks very promising. And I should point out that the toxicity profile was, as expected, quite manageable. So there was a modest increase in diarrhea, but otherwise the toxicities was pretty similar to what was seen with the capecitabine trastuzumab control arm. So a big benefit in terms of efficacy and certainly a manageable increase in toxicity. So I think this is a very promising drug for patients. We also heard about a study of margetuximab, which is another interesting molecule. This is essentially the trastuzumab monoclonal antibody with the Fc portion, which is a constant region of the antibody, modified so that it binds to Fc receptors better. And this is important because it's the binding of trastuzumab to Fc receptors that leads to the immune effects of trastuzumab, both through this phenomenon called ADCC, or antibody-dependent cellular cytotoxicity, and perhaps other immune effectors. So margetuximab was designed to increase the immune effects of trastuzumab, particularly in those patients who have a genetic Fc receptor polymorphism that leads it to be less effective. And this is actually common in about 85% of patients. So the hope was that margetuximab would work better than trastuzumab, but particularly in those patients who have these low affinity Fc receptors. So the trial, which was called the SOPHIA trial, randomized patients with HER2-positive metastatic disease that had been pretreated to either chemotherapy of physician's choice with trastuzumab or chemotherapy of physician's choice with this new antibody overtuzumab. And it did show a modest but statistically significant improvement in progression-free survival, and overall survival was immature with the data was presented at ASCO. At San Antonio in 2019, we had updated data which again showed a modest statistically significant improvement in progression-free survival, and overall survival remains immature, but there is a at least slight trend towards improvement in overall survival of about 4 months that was not statistically significant, but perhaps was even more noticeable in those patients who had these low affinity Fc receptors. So that fits with the anticipated mechanism of action of margetuximab, that it may work better in those patients with these low affinity Fc receptors, which again make up roughly 85% of the patient population. Switching to margetuximab really doesn't have much difference in terms of toxicity profile, with the one exception compared to trastuzumab of that there are more infusion reactions, but these tended to be mild. So I think this is a very interesting drug with a novel mechanism of action, and we're kind of awaiting further data from this trial, particularly to see if the survival benefit becomes more distinct and whether there is a clear difference between the two genotypes of Fc receptors to say that we should only be using the drug in those patients with low affinity receptors, or should it be used in all patients.